Okay, so good morning once again. So for today, we will be talking about the aerobic gram-positive bacilli. So obviously, for gram-positive bacilli, we will be dividing the lecture into two. So the first one is this aerobic. So when you say aerobic, those that can live in the presence of oxygen. And then we also have the other group of gram-positive bacilli, and that would be the anaerobic gram-positive bacilli, which will be on another topic. Okay, so for the aerobic gram-positive bacilli, we will also have several categories later on. So I will be sharing with you what are the different categories. So again, just so to give you a background, whenever we say aerobic gram-positive bacilli, so obviously when you say gram-positive, they stain purple or violet. Okay, and then you'd also notice that they are rod shaped. Okay, so this is an example of non-spore former. Some of them could be spore former, meaning to say they produce spores. And this is an example of non-branching because there are some gram-positive bacilli that are actually branching. So that's the reason why later on we will have several subcategories of this particular lecture. So let's discuss first the general characteristics. Okay. So as what I've mentioned, there are large diverse group of bacteria. So some of them could be spore forming. So again, let's review what we have learned from spores from the during the prelims. So spores are made up of, when you say spore forming, it means that they have endospores. And endospores are important for survival in extreme condition. And to recall, they have calcium dipicolinate as the active substance. And we consider this also as a virulence factor. Okay? So you'd also be able to see branching or non-branching. So later on, I will be showing you the picture of a branching gram-positive bacilli. So they are commonly encountered in the clinical laboratory, microbiology laboratory, and most members are highly pathogenic. Okay, so they are highly pathogenic. Ibig sabihin, they are disease-causing, and some of them are actually being used also in bioterrorism. Okay. So this is the schematic diagram for the presumptive identification of a gram-positive bacilli. So obviously, they're all gram-positive rod. So when you say spore former, you need to say they have endospore. And for those who are spore formers, there's one important genus, and that is Bacillus. And Bacillus may be categorized as beta hemolytic and non-beta hemolytic. So remember that all Bacillus are beta hemolytic except for Bacillus anthracis. And then those that are non-spore former are categorized whether they are branching or non-branching. So these are examples of branching. Okay, so we call them actinomycetes, aerobic actinomycetes. So when you say aerobic actinomycetes, we are referring to the branching gram-positive bacilli. So this includes Nocardia, Gordonia, Chocomorella, Rhodococcus, Streptomyces. But for this particular topic, we will be focusing our discussion on Nocardia. So Nocardia is an example of a slightly acid-fast bacteria. Okay. Under the non-branching, non-spore-forming gram-positive bacilli, We have the Arcanobacterium, Corynebacterium, Erysipelotics, Gardnerella, Lactobacillus, and Listeria. So to classify them, we use a catalyst test. So if you haven't watched the module on Staphylococcus, catalyst utilizes hydrogen peroxide. The catalyst test, in Tagalog, that's agua oxygenada. Based on the principle that the catalase enzyme can hydrolyze hydrogen peroxide into water and oxygen. And because there is oxygen, a positive reaction is the presence of bubbles or rapid effervescence of gas. So those that are catalysts are Corynebacterium and Listeria. So we will also be discussing them. And to differentiate them, we determine whether they are motile at room temperature, particularly at 22 degrees centigrade. And the other one is the bile esculin hydrolysis. So for the bile esculin hydrolysis and motility at room temperature, Listeria is positive while Corynebacterium is negative, meaning to say they are non-motile and they cannot hydrolyze bile esculin. Those that are catalase negative are Arcanobacterium, Erysipelotics, Gardnerella, and Lactobacillus. So we will be focusing ourselves with Erysipelotrix and Gardnerella because they are the ones that are highly pathogenic. Although I will not be discussing Lactobacillus now, but uh, let give me let me give you an idea of what lactobacillus is. So these are capable of fermenting lactic acid, and majority of them are actually beneficial organisms. In fact, one of the species of lactobacillus is Lactobacillus acidophilus, and Lactobacillus acidophilus is also known as the Norderlin's bacillus, and they are important vaginal flora. Because of the presence of Lactobacillus acidophilus, uh, organisms that can cause urinary tract infection will not actually grow. Okay, so it's important part of the vaginal flora. And then also, um, it's beneficial organisms. In fact, we want to have more Lactobacillus casei. Okay, if you remember, if you can recall, um, Lactobacillus casei, Shirota strain is actually found in Yakult. Okay, and whenever we drink this, oops, nawala. So whenever we drink this, um, this makes our this makes our intestine, okay, rich in normal healthy flora. Okay, so going back to our discussion here, so they are classified whether they are H2S positive or H2S negative. So those that can produce hydrogen sulfide. So hydrogen sulfide positive is characterized by blocking in triple sugar iron. So later on, I will be discussing it also. Okay, so our first group of organism are those that are non-spore forming, non-branching, catalase positive. Okay, so we will be discussing first the Corynebacterium. Okay, so Corynebacterium. Corynebacterium is a very diverse group of bacteria. It's a very diverse group of bacteria, and they are found as animal and human pathogens. So some of them are saprophytes. some of them are actually saprophytes. And so when you say saprophytes, what's happening? Uh, Sorry for that. So when you say saprophytes, it means that they actually live on decaying organic matter. So that's what we meant by saprophytes. So most of the fungi are saprophytes, but incidentally, Corynebacterium are also classified as saprophytes. Okay, and as what I've mentioned to you, they are diverse. Sorry guys, problema yung tablet ko. Should end show this card. For example. Okay, so again, so they are saprophytes, meaning to say, they live on decaying organic matter. So majority of them are found in normal biota of both humans and animals. So when you say normal biota means that um, there are there are some species that are normally present inside our body, particularly in the skin and mucous membranes. Okay? And based on 16S rRNA, so this is the genotypic characteristics. So when we say 16S rRNA, we are referring to the genotypic characteristics. They are closely related to Mycobacterium and Nocardia. So that's understandable because Mycobacterium and Nocardia are actually gram-positive bacilli, although both of them are said to be acid-fast. Okay, so that's the general characteristics of Corynebacterium. So Corynebacterium is actually one of the most diverse group of bacteria, and they are highly pleomorphic. So when you say pleomorphic, ito pala, pleomorphic means that they assume a variety of shape. So they assume a variety of shape and they are palisading. So when you say palisading, means that they are arranged side by side. So the term corine is actually a Greek word which means club. Okay. And if you will be looking at the morphology, okay, if you will be looking at the morphology, you'll be able to see a character that has Chinese lettering that resembles Chinese letters. So Chinese letter characters, so that is actually for Corynebacterium. Okay, so this is an example of a special stain for Corynebacterium. And if you will be looking closely, you'll be able to find for Corynebacterium these structures. Tanin nyo? So these structures are called metachromatic granules. But these metachromatic granules will only be seen if you are using a special stain. So metachromatic granules, or sometimes known as volutin, serves as the food reservoir of the bacteria. For Corynebacterium diphtheriae, there's a special name of metachromatic granules. Okay? So the metachromatic granules of Corynebacterium diphtheriae is called Babes Ernst granules. So ito lang yung bacteria na merong babes. Ayan. So sana all, di ba? Ayan. So babes earns granules. Kayo ba? Meron ba kayong babes? Ayan. So buti pa si Corynebacterium, meron siyang babes. Pero yung kanyang babes ay babes earns granules. That's the special name of the metachromatic granules. But take note that metachromatic granules will only be seen not through gram staining, but only through a special stain. Okay? So there are two categories of Corynebacterium. So we have the non-lipophilic and the other one is lipophilic. So when you say lipophilic, so let's dissect the word. Lipo refers to fat or lipid. Philic is actually love of. So when you say lipophilics, they love fats. In fact, for lipophilic organisms, if lipids are in culture, the growth is enhanced. Okay, katulad nito. So lipids are found in culture, and that's the reason why the growth is enhanced. So they are fastidious, slow growers, and requires 48 hours of incubation before the colonies like this will appear. So let's dissect again, or let's recall what we mean by fastidious. So when you say fastidious organisms, these organisms would need enriched medium for them to grow. So that's what we meant by fastidious. Means that they would require enriched medium. So when we say enriched medium, meaning to say you have added additional nutrients to the culture media, and these additional nutrients can be in the form of blood, for example, lipids. Okay, so that is for lipophilic microorganisms such as Corynebacterium. So they are slightly curved gram-positive rods. So that's the reason why they are pleomorphic. They are unparalleled and slightly wider ends. Okay, so they are described as the club shape. Okay, and if the organisms appear like this, we call them dipteroid-like. So they are dipteroid-like. So yung club shape, ito yon. Kaya siya tinawag na club shape. However, this particular photograph is stained using a methylene blue, okay, from the pie slant culture. Pie slant is an example of medium that contains lipid. So that's the reason why you will see them that the growth is really enhanced. Okay, so are they clinically significant? Yes, they are clinically significant. And one of the most clinically significant species is Corynebacterium diphtheriae. So take note how we spell diphtheriae. This is spelled like this. Okay, particularly whenever we are referring to the disease, to the scientific name. So Diphtheria. Okay, so it causes diphtheria. And diphtheria is an infection of the upper respiratory tract. And usually, you will see the presence of pseudo membrane formation. So there is a pseudo membrane formation. Okay? And it is an example of throat without pseudo membrane. And this one is an example of throat with pseudo membrane. So other species may cause infections, particularly in immunocompromised hosts, because these are people with very weak immune system. Okay, so let's talk about the virulence factor. So Corynebacterium diphtheriae is an example of toxigenic bacteria. Okay, so let me give you an example of other toxigenic bacteria. So aside from Corynebacterium diphtheriae, other examples of toxigenic bacteria are Clostridium tetani, which can cause tetanus, and tetanus toxin is the reason for that. Okay. And then we also have um, aside from Clostridium tetani, other examples of toxigenic bacteria is Bordetella pertussis. Okay, so yung Bordetella pertussis can cause whooping cough. Okay, so Corynebacterium diphtheriae is an example of toxigenic bacteria. So the toxin is worldwide in distribution and rare in places where vaccination program exists. Okay, so that's the reason why it is very important that we immunize our children. So is there vaccination? Yes. So the vaccination is actually being given in the form of toxoid. So pag sinabi nating toxoid, we are referring to attenuated toxins. We are referring to attenuated toxins or the weakened toxins, and this is usually being given DPT, diphtheria, pertussis, tetanus. So if we give this to our baby, if we immunize our babies with DPT, we are preventing them to get diphtheria, pertussis, which is caused by Bordetella pertussis, and Clostridium tetanus. Okay? So without DPT, then chances of getting diphtheria would be very high. So what makes Corynebacterium diphtheriae virulent? It is because of the presence of the diphtheria toxin. And the reason why the organisms can produce diphtheria toxin is actually due to the presence of the tox gene. Okay? So there are two types of Corynebacterium diphtheriae. Okay? Those without tox gene, they are considered to be as a virulent. But those with tox gene, kung meron na silang tox gene, then we consider them to be the virulent Corynebacterium. And Corynebacterium diphtheriae will get tox gene if they are injected by lysogenic bacteriophage with tox positive or tox gene. Katulad nito. So this is an example of a bacteriophage. So if the virulent bacteriophage will be injected with the tox gene, then the bacteria will now acquire the tox gene. And that will make this bacteria virulent. Okay? So it says here that only toxin producing bacteria can cause diphtheria. If, for example, the bacteria are not capable of producing do not have toxin, then they are a virulent. So toxin production is inversely proportional to iron availability. Okay, so the less iron, then the more toxin production will happen. So toxin is antigenic, meaning to say, we can use antibodies to block or neutralize the toxin effects. But if we do not want to get infected, it is very important that we immunize our children with the toxoid DPT. Because if we give them DPT, then we prevent diphtheria. So there's no need for us to immunize, to neutralize the existing toxin in the present in the body. So prevention is better than cure. So that's what they said. So this is an example of electron micrograph of bacteria and numerous bacteriophages trying to inject, okay, the genetic materials into the bacteria. So in this case, that is the bacteria diphtheria toxin. So it occurs in strains infected with lysogenic beta bacteriophage, which carries the responsible toxin. So non-toxigenic strains, when we say non-toxigenic strains, we are referring to the virulent strain, can be converted to tox positive whenever they are being injected with the appropriate bacteriophage. So the diphtheria toxins are made up of two fragments. Okay, so fragment A, this is the active fragment, which can inhibit protein synthesis. And then fragment B are the ones that will bind to the specific cell membrane receptor. So, so fragment B, therefore, is responsible for the entry of the toxin to the specific cell because it mediates the entry of fragment A into the cytoplasm. Okay, so again, these are the two fragments of the toxins. So, what will happen if you get diphtheria? So how do you get diphtheria? You can get diphtheria by means of droplet spray. So meaning to say respiratory droplets. So by wearing mask, whenever you interact with someone who has diphtheria, then you can prevent yourself from getting infected. Or also through hand-to-mouth contact. So those people who are unimmunized, meaning to say those who did not receive the vaccine, will be more susceptible to get diphtheria. Okay? So the incubation period would last for about 2 to 4 days, so very short. And then at the start, the person will start to have a low-grade fever characterized by malaise and mild sore throat. So local toxin production will cause tissue necrosis and starts at the tonsil and spread downwards into the larynx and trachea. And this is somewhat dangerous because the presence of pseudomembrane will obstruct breathing, proper breathing. So this is an example again of a pseudomembrane. So it is composed of bacteria, lymphocytes, plasma cells, fibrin, dead cells, which covers the uvula, tonsils, and palate. So without antibiotic treatment, this particular pseudomembrane may even reach the trachea, the larynx, and the trachea. And we know for a fact that our trachea is our windpipe. And people with pseudomembrane will try to cough it out, and as they try to cough it out, there would be forceful coughing, and this may even cause pseudomembrane to be torn away from the mucous membrane itself, and there would be bleeding as well. Okay, so that is Corynebacterium diphtheriae. So we do not want our children um, to get infected with it. So that's the reason why it's very important to have proper immunization. Okay, aside from the respiratory infection, the systemic effect is equally dangerous because sometimes toxin is absorbed in the bloodstream and carried systematically. So this condition is called toxemia. And toxemia would have a drastic effect because it will affect our kidneys, heart, and nervous system. So if the nervous system, for example, is affected, there would be demyelinating peripheral neuritis, which could possibly result into paralysis. If heart is affected, there could also be cardiac failure, and this could be fatal as well. If kidneys are affected, there would be renal failure, so it would require patients to be in dialysis, and if not, it could also be fatal. So that's the reason why we do not want to get toxemia. So as much as possible, we should immunize our children. Okay? So that they do not have to suffer from this particular effect of Corynebacterium diphtheriae respiratory infections and the consequences. So there's also a chance of children or people getting cutaneous infection. So it is actually it occurs much less often than respiratory. So it happens when the toxin is absorbed systematically, and it will go to the cutaneous area. So there would be non-healing ulcer with dirty gray membrane. So it is not usual in developed countries such as in the United States, but I think the Philippines we have also minimized Corynebacterium diphtheriae because of our very good campaign with the vaccine. Okay, so let's talk about the microscopic examination. So as you can see, if stained using gram-positive rod, they are arranged in palisade and they are club-shaped. And some of them may resemble letter V, such as this one, or some of them may resemble letter T, yung iba nga letter Y. Okay, but if we're going to use the methylene blue stain, which is the special stain, you'll be able to see the beaded appearance, such as this one. And they possess the metachromatic granules, which I have already mentioned to you in the introduction. So that particular metachromatic granules, or sometimes known as volutin, okay, the specific name of volutin for Corynebacterium diphtheriae is called the Babe's Earns granules. So again, this particular Babe's Earns granule is composed of polymerized phosphates, which serves as the food reserves of the Corynebacterium diphtheriae. Okay, so again, so this is the gram stain, and then this is the methylene blue stain of the diphtheroids. Okay, so very intense metachromatic granules are obvious in this particular type of stain. So let's talk about how do we culture Corynebacterium diphtheriae. So take note that they are facultative anaerobe, meaning to say they are really aerobic by nature but can still live even in the absence of oxygen. And if you really want to see the metachromatic granules or the Babe's Earns granules, you need to culture them in loffler serum agar. This is a lipophilic medium, which would enhance the formation of the metachromatic granules. In sheep's blood agar, you'll be able to see a very small zone of beta hemolysis. So beta hemolysis means that there is a complete lysis of RBC. So there is a complete lysis of RBC. And another important medium is the cysteine tellurite blood agar. This is known as the modified Pinsdale medium. It is selective and differential at the same time. Why selective? Because the reason why we call it selective, because it can inhibit other bacteria from growing. It would only allow Corynebacterium to grow. And why is it differential? Because we can actually differentiate three strains or three varieties of Corynebacterium diphtheriae. Because of the CTBA, we can categorize them as to mitis, intermedius, and gravis. So that is the reason why CTBA is selective, and at the same time, they are differential. Okay, so when culturing Corynebacterium diphtheriae, you'll be able to see black colonies with brown halos around the colonies. So this is the Loeffler's medium. So this one is very high in lipid content. So what's the importance of this medium? They can enhance metachromatic granules formation. And you'll be able to see organisms in pleomorphic formation. Okay? And this is the blood agar plate, the sheep's blood agar plate. So you're supposed to see a very small zone of complete hemolysis in this medium. And this is now the CTBA. So you'll be able to see black colonies with halos surrounding it. So it is selective and differential. Selective because it only allows Corynebacterium to grow, differential because we can differentiate three important characteristics of Corynebacterium diphtheriae. So we can differentiate them as to gravis, meatis, and intermittus. Okay? So when you say gravis, the colonies appear to be this one, large, flat, dark gray, irregular striated, daisy head colonies. So this one is negative in hemolysis, but positive in starch hydrolysis. So the reason why we call it gravis, because this one will cause the most severe form of diphtheria. Then the third type would be the meatis. So the meatis is characterized by small, black, glossy, convex, translucent that appears like the fried egg appearance. So the meatis is positive also for hemolysis, but negative for starch hydrolysis. So the meatis will result into a mild form of diphtheria. And then we also have the intermedius characterized by um, small, flat, dry gray, black colonies on CTA. So this is the intermedius. And obviously, they are negative in hemolysis and starch hydrolysis and will result into an form, intermediate form of diphtheria. Okay, so these are the three types of colonies of Corynebacterium diphtheriae as shown in cysteine tellurite agar or the modified Kinsdale medium. So how do we identify them? Medically important species are catalase positive and they are non-motile. So we can confirm them by fermentation reaction and urease production is supposed to be negative. But toxigenicity testing will determine if the Corynebacterium diphtheriae that we have isolated are capable of producing toxins or not. So there are two types of toxigenicity testing. Okay, so it could be the in vivo or the other one is the in vitro. So an example of in vivo is the mouse virulence test. And the example of in vitro is the ELECTR test. So ELECTR test is an example of immunodiffusion test. Okay. And of course, this will tell us whether the Corynebacterium diphtheriae that we have isolated is indeed toxigenic. But of course, the gold standard is molecular technique. So we can do PCR and look for the presence of the tox gene. But this one is expensive. Okay. So just so to give you a background. So if the colonies on Tinsdale medium will produce brown halo, we do the gram stain. Okay. So if it's positive in brown halo, then we do the gram stain. And if it's gram positive, they could be diphtheroids. So to differentiate them, we do urease test. And they should be negative because Corynebacterium diphtheriae is negative for urease test. And then the other one, you do the starch hydrolysis or starch fermentation. If it's positive, then it could be Corynebacterium ulcerans. And then if it's negative, it could be Corynebacterium pseudotuberculosis. Okay, so this is how we identify Corynebacterium diphtheriae and other species of Corynebacterium. Now, speaking of in vivo test, we have the mouse virulence test. Okay, so here you need to have two sets of animal. Okay, so we have the test animal and then the control animal. So for the test animal, we give the test animal the suspected bacterial cell suspension. So this is a suspected bacterial cell suspension. For the control animal, we give this bacterial cell suspension plus the diphtheria antitoxin. So we inject. If the test animal is dead, and the control animal is alive, meaning to say you have isolated a toxigenic bacteria. So the reason why the control animal is alive is because there is an antitoxin that can neutralize the toxin being produced by Corynebacterium diphtheriae. If the test animal is alive and the control animal is also alive, meaning to say the bacteria that you have is non-toxigenic. But if the test animal is dead and the control animal is dead, the reason why these animals die is because the toxicity is not due to diphtheria. It could be due to other diseases. Okay, so animal inoculation test is rather tedious and you need to acquire clearance from the ethics committee before you can actually do it because it involves live animals. So that's the reason why most of the laboratories are just doing the ELECT test. So the ELECT test is just made up of a strip. And as what I've told you, the principle of ELECT test is called immunodiffusion. So the principle of ELECT test is just immunodiffusion. So all you need to do here is to inoculate bacteria. So these are bacteria 1, which is a positive control. And then the bacteria 2 is the unknown. This is now your test organism. And then bacteria 3 is the negative control. And number 4 is another unknown. Okay, so we will just be comparing the result for number 2 and 4. Because number 2 is an example of toxigenic bacteria. And then number 4 is an example of non-toxigenic bacteria. So after we inoculate the bacteria, we add, okay, strip on top of this. Okay, so the strip is actually made up of antitoxin impregnated filter paper strip. Okay, so what would be the positive result? So the positive result would be the lines of identity. So this is an example of the lines of identity. So uh, take note that between number 1, which is the positive control, and this is toxigenic, there is a semi-arc formation, which is, of course, a line of identity. So this line of identity is called the precipitin line, which is not present for non-toxigenic bacteria. So ELECT test is actually much simpler as compared to the animal inoculation test. So that's the reason why it's actually being done more in the laboratory as compared to the animal inoculation test. But again, the gold standard is still PCR. So simply look for the presence of the toxin, but not all laboratories would be capable of doing such. So this particular table will tell us the different identification scheme for Corynebacterium, different species of Corynebacterium. So take note that Corynebacterium diphtheriae will have a halo growth in Tinsdale. So are they lipophilic? So the results would vary. Um, Also in beta hemolysis, the results would vary because there are 3 types, remember? And then they're positive in nitrate reduction, in alkaline phosphatase, glucose fermentation, and maltose fermentation. Treatment. So how do we treat diphtheria? So we can treat diphtheria by administration of antitoxin. So the purpose of administering antitoxin is to neutralize the toxic effect. Okay? So we eliminate after that we eliminate the focus infection and prevent the spread of organisms by giving antibiotics. So penicillin is the drug of choice. Erythromycin is also being used for penicillin sensitive individuals because there are some individuals who might be allergic to penicillin. The problem with antitoxin is that antitoxins are actually being produced in horses, which means that there are times that people would actually have um, toxic reactions toward or allergic reaction towards the horse antitoxin. So there are other species of Corynebacteria. So one of them is Corynebacterium amiculatum, which is common on skin and most frequently recovered species. It is an opportunistic infection which can infect our prosthetic joints. It may cause sepsis and endocarditis. It is seen as a nosocomial, so meaning to say hospital infection, and it may be an opportunistic infection for patients who are immunocompromised. And then we also have Corynebacterium JQ. Okay, so in some texts they call it JK. Okay, so infections from catheters or prosthetic devices. So if it's in prosthetic device, which is for example, you have a prosthetic valve, so it may even affect your heart, causing prosthetic valve endocarditis. And then Corynebacterium pseudodiphthyricum is another a rare opportunistic infection, and they are mostly frequently associated with respiratory tract infection. Then we also have Corynebacterium pseudotuberculosis. Usually we get it if we have contact with sheep. So you notice that this particular sheep is infected because it is related with dermonecrotic toxin. Nalobat na yung excuse me. Yan. And then Corynebacterium striatum, another rare infections, but it is part of the normal flora. And then Corynebacterium ulcerans. So interestingly, it may cause mastitis in cattle. Yung mastitis is actually um, the milk glands. Okay, so the udders of the cattle are actually affected in this particular infection. So we can get it if we have come into contact with this animal or if we have drank unpasteurized milk. And then, incidentally, Corynebacterium urinalicum is said to be positive with urease test. Highly positive. Within minutes, it will yield a dark pink result, and it is considered to be as a urinary pathogen. Okay, so let's now discuss identification of Corynebacteria. We speciate if we have isolated it from normally sterile site. Okay, so predominant organisms from properly collected specimen. So ano ba yung normally sterile site? Our bladder is supposed to be sterile. Our blood is supposed to be sterile. Okay. So urine sample, if soil isolate is greater than 10 is to 4 per ml of organisms and not greater than 10 to 5 if they are predominant. So we have to speciate. Meaning to say, when you say speciate, we have to identify up to the species level. Okay, so that ends our discussion with Corynebacteria. Okay, so again, let me just explain to you. So this is the club-shaped pleomorphic bacteria, and this is a blood agar plate, and you can actually see small beta hemolysis in here. I only have one slide for Rosia. This one, which belongs to the family Micrococci, Rosia dentocariosa. It's a normal oral flora, but may cause endocarditis. So again, they always say that if you have unhealthy mouth, the chances of you getting heart disease would be very high. So group G has been associated with bacteremia, endocarditis, food infection, and eye infection. Okay, so let's now proceed to another interesting gram-positive bacilli. And this time, we'll be talking about Listeria monocytogenes, a gram-positive, non-sporulating cocobacilli. Wow, what is a cocobacilli? Cocobacilli is a plump bacillus. Plump, matataba. And its because they are plump, sometimes they may even appear like cocci, but actually they are classified as bacilli. So they are widespread in nature and primarily infect animals. So actually, um, people who are immunocompromised are actually more predisposed to have it, and even neonates. But we can be exposed to listeria, so we call it listeriosis. Okay, and so our exposure to listeriosis is very limited unless we actually consume dairy products because these organisms can even grow at low temperature. And we have consumed processed meat, okay, that are not hygienic, that are not hygienic when it comes to preparation. So these are conditions where we can get listeria or listeriosis. So there are several virulence factors of Listeria monocytogenes, and among these, the most noteworthy to mention is hemolysin. So again, when we say hemolysin, they can actually cause lysis of RBC. But there's a very specific name of hemolysin for Listeria monocytogenes, and this is the listeriolysin O. Listeriolysin O can damage phagocytic membrane and prevent killing of the organism. So this is highly virulent for Listeria monocytogenes. And from that, we have catalase, superoxide dismutase, phospholipase C, and surface protein P60, which can also induce phagocytosis. So can adults be infected with listeria? Actually, yes, and it's actually more dangerous to have listeria monocytogenes infection if someone is pregnant. So in adult, usually we get it as a result of consumption of unhygienic food or food that has been prepared unhygienically. So for these particular individuals, they would likely develop diseases from penetration of the intestine, and eventually it will spread systematically. So pag sinabing systematically spread, meaning to say blood through various organs. And if it's CNS, then it may cause meningitis. And people who are most at risk are immunocompromised or elderly. And from blood, it may even go to our heart and may cause endocarditis. For pregnant women, it is actually dangerous. So pregnant women will usually feel mild flu-like syndrome, and it could be fatal to fetus, unfortunately. And it may even cause premature labor and septic abortion. So that's why pregnant women should be very careful in things that they are consuming because most of these are found in food, listeriosis. Okay, so listeriosis infections in newborns and immunocompromised hosts. So it may even cause diseases in newborns, so neonates. So how did these neonates will have listeriosis? Because of the vertical transmission. So when we say vertical transmission, this refers from mother to fetus through intrauterine transmission. So it will result to sepsis, and eventually, when you say sepsis, it is in the blood, right? And then it will go to CNS. So there would be meningitis. So there are two types of onset, huh? The early onset and then the late onset. The early onset would have a high mortality rate, but the late onset would have lower mortality rate. For immunocompromised hosts, it may cause infection in the CNS and even in the heart. So endocarditis naman yung tawag doon. So how do we get listeriosis? Diseases in otherwise healthy individuals would involve eating contaminated food. So those food that are at risk of having listeriosis are those that are rich in milk, in mga dairy products, or even processed meat. So, for example, cheese, ice cream, coleslaw, these are made rich in dairy products. So, di ba, for example, coleslaw is actually rich in mayonnaise. And then hot dogs, lunch meats, and chicken, these are processed meat. Okay, so these are the food that would be more prone of getting Listeria monocytogenes. So possible effective treatments are penicillin, aminoglycosides, and macrolides. So under the microscope, Listeria monocytogenes is characterized by gram-positive, non-spore-forming Bacilli. So if we're going to enlarge, so there are, but because Bacilli are supposed to be like this, cocci are supposed to be like this, but this is somewhere in between. So that's the reason why we call them Bacilli. So my, but if we're going to do this is from the direct smear. Ah. So when we say direct smear, smear coming from clinical samples. This is what we call direct smear. But this one is called microscopic, the culture. So ibig sabihin, these are smears coming from culture media. If the smear came from culture media, if the smear came from the culture media, they are characterized by single short chain palisades. So palisades means say side-by-side arrangement and can resemble streptococcus in coccoidal form. Okay, so this is how they look under the microscope. So again, this is a smear from the culture. So this is the Coccobacilli. This is the direct smear. And then this is the smear from the culture. So they are cocci na, no? Para na sila mga cocci. They can grow well on blood agar plate. This is the SBA. And then the chocolate agar plate. But they would need a slight increase of CO2. So in blood agar plate, for example, they are characterized by small, round, smooth, translucent, and they produce also a narrow zone of beta hemolysis that is almost similar to group B streptococcus. So if you remember group B streptococcus, they are streptococcus agalactiae. I hope you have watched it already. And cold enrichment is also possible for them. So when you say cold enrichment, you allow them to grow at 4 degrees centigrade before plating them in primary culture medium. Okay, so that is the cultural characteristics of Listeria monocytogenes. So here again is the beta hemolytic, okay, that is similar to Streptococcus agalactiae. So incidentally, Streptococcus agalactiae is also positive with CAMP test. What is a CAMP test? CAMP test is Christie, Atsky, Munch, Peterson. Okay? For Streptococcus agalactiae, the result in CAMP test is called arrowhead hemolysis. But for Listeria monocytogenes, the result is called block hemolysis. So this is an example of the block hemolysis. No? So what is a CAMP test? CAMP test is actually the perpendicular growth of Listeria monocytogenes beside the colonies of S. aureus. So this is Staphylococcus aureus, and then this is Listeria monocytogenes. And notice that if they are if they are together, so the junction in between them, this is the block hemolysis. Okay, so that is the characteristic of the CAMP test for Listeria monocytogenes. So if you want to know more about the arrowhead hemolysis, so the I actually discussed this one under Streptococcus, but for Listeria monocytogenes, it is called block hemolysis. Okay, so Listeria monocytogenes is positive in catalase test. So in catalase test, this is a hydrogen peroxide. So there would be rapid effervescence of gas or presence of bubbles, and that is a positive result. So again, do not use colonies from blood agar plate if you are to perform catalase test, because it will result to false positive result, and blood would have naturally occurring catalase enzyme. And separate hydrolysis is characterized by purple color. So that is another reaction for Listeria monocytogenes. And then motility at room temperature, there would be the umbrella type motility. So this is the umbrella type motility. So how do we test it? So usually we have a semi-solid medium. So we use an inoculating needle and we stab halfway. And after we have stabbed it, after 24 hours, you would notice that the growth would be like this. So this is the umbrella motility. And then if you will be preparing it in hanging drop, meaning to say, slide plus the culture medium with liquid suspension, they will tumble. So tumbling motility. So take note that tumbling motility and umbrella type motility is only possible if incubated at room temperature. So that's why at 25 degrees centigrade, not at 35 degrees. So Listeria monocytogenes is catalase positive, positive in bile esculin hydrolysis, positive in motility, positive in beta hemolysis, and can grow in the presence of 6.5% sodium chloride. So this test is called the salt tolerance test. Okay, so when compared to other gram-positive bacteria. So it's another example of a positive in umbrella motility. This one is negative for umbrella motility. And then the CAMP test, which I've already explained to you, it's produced a black type of hemolysis. In contrast with Streptococcus agalactiae, which is the arrow shape, this one is the black type of hemolysis. Okay, so yeah, I think I've shown this already. Okay, so we have now come to other group of gram-positive bacilli. So this time we'll be talking about the non-spore forming, non-branching, catalase negative bacilli. So first on our list is Erysipelotrix russio-Patriae. Okay, so you might be wondering, what is russio-Patriae? Russio refers to red. Patriae refers to disease. So literally, the term russio-Patriae refers to red disease. Okay? Because later on, you would notice that the main disease is called erysipeloid, and it is characterized by a red rash. So that's why the term russio-Patriae, which is found worldwide, It can be a commensal or a pathogen in a wide variety of vertebrates and invertebrates. And incidentally, usually yung mga pigs, yung nag pig serves as the reservoir of erysipelotrix rhizospathiae. Human cases typically arise from occupational exposure, meaning to say people who handle fish and animal products are mostly at risk of getting erysipelotrix rhizospathiae. So how do we get this? Through cuts or scratches in our skin. So we can get erysipelotrix rhizospathiae so through cuts or scratches on our skin. So the organism is resistant to salting, pickling, and smoking. Meaning to say, if kahit if smoked fish or smoked meat may still have erysipelotrix rhizospathiae. Okay, so ano ba yung example ng smoked meat? Ham. So if ham is prepared from pig with erysipelotrix and then you will be eating hams raw na smoke lang, then there's still a chance that you might still get erysipelotrix. And the organism survives in environmental sources such as water, soil, and plant material. So this is an example of erysipeloid. So you see, it's not really a rash, but it's more of a painful swelling usually found on the hands or fingers. So medyo nagkaroon lang ng, di pala rash, so I stand corrected. So parang nagkaroon ng red discoloration. So it's a self-limiting localized infection at the site of inoculation. Meaning to say, siguro nagkaroon ng cut dito or ng scratches, then nakapasok dito yung erysipelotrix rhizospathiae, which could heal within 3 to 4 weeks. So it's another example of erysipeloid. So erysipeloid is a local infection, which would heal by itself. But what we do not want to get is a systemic infection. Meaning to say, from the local infection, it goes to the circulation. So there would be what? Septicemia. And it is associated with endocarditis. It may occur in those who have had valve replacements. Or there would be a diffuse cutaneous infection. So it manifests as an exacerbation of erysipeloid lesion. So again, from a local infection, then it will now diffuse. So systemic infections are uncommon. And if we do not want to have systemic infection, it is very important that at the early onset of erysipeloid, so for example, nakita na meron ng rashes or meron ng, reddening of the skin, then we can treat it with penicillin. And if the patient or if the bacteria is resistant to penicillin, then we can actually go to vancomycin. Okay, so under the microscope, they could be gram variable. As you can see here, some of them may be purple, some of them may be pink. So that's what you call gram variable. So they are thin, rod-shaped, and pleomorphic, and may actually appear as long filaments, such as this one. And they are arranged sometimes singly or in short chain or V-shape, such as this V-shape. Okay, so that is how erysipelotrix rhizospathiae appear under the microscope. So another V-shape. Okay, so this is another example of gram stain of erysipelotrix rhizospathiae. For cultural characteristic, CO2 is required, so they can grow on blood agar plate. The colonies are usually non-hemolytic. So when you say non-hemolytic, it means that they are gamma hemolysis, meaning to say there's no hemolysis. So after 24 hours, they are pinpoint. But after 48 hours, the colonies may appear as smooth, form, transparent, glistening convex with entire edges, such as this. Okay, so that is how erysipelotrix rhizospathiae occur in culture. After 48 hours, again, So they may now appear as large colonies, rough flat with irregular edges. Colonies appears to be alpha hemolytic now, and and subcultures from both should be inoculated daily to SPA. So from broth pala to, from broth should be inoculated daily to sheep's blood agar. So ano ba yung purpose ng broth culture? And then broth culture is the enrichment. And then pag sinabi natin plate culture, this is an example of plate culture. This is the primary enriched medium. So blood agar plate or the sheep's blood agar is an example of enriched medium. Ano naman? So there is a so to compare, this is the Listeria. Sorry. Yeah, this is the Listeria, and then this is Erysipelotrix. So this culture would appear after 48 hours. So nakikita nyo na that it will start to show alpha hemolysis. So how do we know if it's alpha hemolysis? Alpha hemolysis, alpha hemolysis means that there is an incomplete hemolysis. Kasi when we say beta, that is the complete hemolysis. Gamma hemolysis means no hemolysis. But alpha hemolysis is incomplete hemolysis. And it is characterized by greenish brown. So the blood agar plate would have a greenish brown surface. So this particular greenish brown surface is an example of alpha hemolysis. Okay, so other identification methods. So they are either aerobic or facultative anaerobic. They are catalase negative, and they can produce blackening on triple sugar iron. So this is the triple sugar iron. So if inoculated in triple sugar iron, there is a blackening. So the blackening in triple sugar iron represents that there is a hydrogen sulfide production. So if there's a hydrogen sulfide production, so that's another characteristics of Erysipelotrix russopathiae. They are Vodge-Proskauer negative, but if we're going to stub them in gelatin agar, it would appear like a test tube brush. So this is the test tube brush pattern. So this particular test tube brush pattern is the characteristic of Erysipelotrix russopathiae in gelatin agar. Then of course, Malditov is another sophisticated way of identifying them. Okay, matrix assisted laser desorption ionization time of flight. So again, to compare Erysipelotrix with other gram-positive bacilli, Erysipelotrix russopathiae is catalase negative, non-motile. They cannot hydrolyze bile esculin, but they can ferment glucose. They can produce hydrogen sulfide in triple sugar iron. The only gram-positive bacilli in this list that can produce hydrogen sulfide. Some of them are, first 24 hours, they are gamma hemolysis. But after 48 hours, becomes alpha hemolysis. They cannot reduce nitrate, and they are negative in urease. So in summary, that's the results of Erysipelotrix russopathiae. So other non-spore forming, non-branching, catalase negative are Arcanobacterium and Truperella. Okay? Truperella. Truperella, sa true lang. Okay, so some members of this genus were Arcanobacterium, so some of them were actually moved to Truperella. So they are primary pathogens that can infect humans, and both of them are catalase negative. So under the Truperella, we have, under this particular group, we have Arcanobacterium hemolyticum, Truperella pyogenes, and Truperella bernardii. So, so let's talk about the Arcanobacterium hemolyticum. So this particular bacteria has been recovered from patients between 10 to 20 years old. So usually yung mga patients ito are actually complaining of pharyngitis. So infection can be mild or severe and usually clinically indistinguishable from that of beta hemolytic streptococcus. So akala natin meron tonsillitis yung patient, may pharyngitis yung patient. So most of the doctor would jump into conclusion that it is caused by streptococci. Kasi nga, bibihira lang naman talaga ma-encounter yung bacteria na Arcanobacterium hemolyticum. So it is associated with soft tissue infections, may cause sepsis, endocarditis, and other infections. So as you can see, they would produce small colonies and sheep's blood agar, and then they are beta hemolytic 24 to 48 hours of incubation. So, paano natin nalaman na beta hemolytic na sila? Diba? Again, when you say beta hemolytic, it means that there is a complete hemolysis. Means that there is a complete hemolysis. And when we say complete hemolysis, you'd see colorless zone. So you will be able to see colorless zone of hemolysis. So that's what we meant by complete hemolysis. So black opaque dots are actually observed when the colonies are scraped away. So kapag kumuha tayo ng colonies on sheep's blood agar, there would be black opaque dots. So pitted agar beneath the colony has also been reported. So pitted means that there's a hole. Gram-positive, there are gram-positive rod pleomorphic with sometimes there are rudimentary branching. So lipase test and lecithinase test is another test that will yield positive result. And sometimes their CAMP inhibition is also seen. And the treatment of choice is erythromycin or for those bacteria that are penicillin resistant. So this is the Arcanobacterium hemolyticum. So some of them would be branching. So this is the blackening. So if the colonies are being scraped away. Truperella. So let's talk about Truperella. So there are two important species of Truperella. So these are Truperella pyogenes and Truperella bernardii. So under the Truperella pyogenes, okay, so there are animal pathogens of cattle, okay, and they may a rare cause of sepsis and wound infection, okay, for humans who have had contact with this particular infected cattle. On the other hand, Truperella bernardii is a rare cause of infection. But if someone is infected with Truperella bernardii, we would expect them to have bacteremia, wound infection, urinary tract infection, or even septic arthritis. So these are the possible infections that can be caused by Truperella bernardii. Okay, so another bacteria that we will be discussing here is Gardnerella vaginalis. Okay, so Gardnerella vaginalis is a short pleomorphic gram-positive rod, or sometimes they are cocobacilli, and sometimes they are actually gram-variable. So Gardnerella vaginosis is actually can cause bacterial vaginosis in female. And if female has vaginosis, they would have discharge coming out from the vagina that is foul in smelling. This is a malodorous discharge and usually seen in vaginal pH of greater than 4.5. So, in order for the pathologist to diagnose Gardnerella vaginalis or bacterial vaginosis, they would need to observe for the presence of clue cells. Okay, so what are clue cells? So do not forget about clue cells. These are mostly being asked in board exams. So clue cells are large epithelial cells with gram variable cocobacilli clustered in the edges. So if you will be enlarging this one, so you'd see bacteria at the edge of the epithelial cells. So this particular structure, so we call it the clue cells. Okay, so you need to have a very good microscope for you to see. So another example, okay, of bacteria on the edge of the epithelial cells. Another example. So they're very small. Okay, it will really take a keen eye for us to see bacteria on the edge of the epithelial cell. So that's what you call clue cell, and that is actually a characteristic of Gardnerella vaginalis. So pathologists would usually do pap smear, okay? But we can actually do, and then once they've um, analyzed the smear, then they can actually categorize it into four criteria. So they call it AMCEL's clinical criteria, okay? So these AMCEL's clinical criteria for the diagnosis can be used if three of the four criteria below are found. Ano ba yung 3 out of 4 na to? Number 1, homogeneous thin white discharge that smoothly coats the vaginal wall. So they would have to examine. So this would be the job of the gynecologist. And then gynecologist will send the sample in the lab. And then in the lab, they would need, we will be preparing the smear and look for the presence of the clue cells. So this is the second criterion. And then the third criterion is we have to measure the vaginal fluid. We have to measure the pH. If it's greater than 4.5, then the chances of them getting Gardnerella vaginalis would be very high. And of course, the fishy odor of the vaginal discharge before or after the addition of 10% potassium hydroxide with test. So unfortunately, we have to smell it. So it has to be fishy odor. Okay, so 3 out of the 4 criteria. So DNA hybridization probe test is the molecular testing for the Gardnerella vaginalis. So cultural characteristic under the sheep's blood agar or chocolate agar, they need to be treated or they need to be enhanced with 5 to 10% carbon dioxide, meaning to say they are capnophilic. And they need to be incubated between 35 to 37 degrees. So you'll be able to see human blood bilayer twin agar. So this is the culture medium of choice. In human blood agar culture, so you would expect them to be beta hemolytic. So again, they are colorless zones. So that's the reason why they are classified as beta hemolytic. So for people or for women that has or that have Gardnerella vaginalis, so the treatment of choice would be metronidazole and clindamycin. And incidentally, if you remember our parasitology, metronidazole is also the treatment of choice for Trichomonas vaginalis kasi both of these infections would result into a foul-smelling fishy odor that would come out from the vaginal fluid. Okay, so this is the Nugent scoring system for Bramstein vaginal smear. Okay, so what does it mean? This will interpret whether the vaginal smear would have the normal flora or pathogenic bacteria. What do we need to have is, of course, the lactobacillus. Lactobacillus, if you remember, I've introduced this to you a while ago. Lactobacillus acidophilus is a normal vaginal flora. This is the Dauder-Lanes. This is the Dauder-Lanes bacillus. This is what we need. Hindi pala, nakisada yan. No? This is what the women need in the vagina. Okay? So, and then for gram variable bacilli, so we have the Mobulinthus morphotypes. Lactobacilli is the Baxi gram-positive bacilli. Mobulinthus, Mobulinthus is the curved gram-negative bacilli. What the women do not need in the vagina are the Gardnerella and Bacteroides. So both Gardnerella is gram variable and Bacteroides is the gram-negative short bacilli, and it is said to be an aerobic. So there would be scoring system. So if it's +4, so it's called a 0, 1, 2, 3, 4. So if you are able to see pleomorphic gram-negative, small gram-negative short bacilli, this is the scoring system. For Mobulinthus, this is the scoring system. Okay. Mobilink. So if your score, and then we add the total score. So you add the total score. If your score is between 0 to 3, that's a good thing because it means that we are referring to the normal vaginal microbiota. For it, if the score is 4 to 6, indeterminate, so it means that the patient may have bacterial vaginosis. 7 to 10, bacterial vaginosis. So if it's bacterial vaginosis, then the patient would need to drink metronidazole as or clindamycin. Okay, so we are now discussing another topic. So this time we will be discussing the non-spore forming branching actinomycetes. Okay, so again, our representative organisms here is nocardia. So as you can see, these organisms are branching. Kaya nga, yung actinomycetes are sometimes called fungi-like bacteria. Okay? Because they are branching. So, nocardia is aerobic, gram-positive, beaded bacilli. Okay, so beads are not spaced at consistent interval. They are partially acid-fast. Kasi the true acid-fast is Mycobacterium. So slight acid fast is nocardia. So colonies and microscopic morphology of the infections resemble the fungi. Kaya nga, nocardia is an example of a fungi-like bacteria. So this is the gram stain. So in gram stain, if it's gram positive, it's supposed to be purple or violet. Okay? And then this is the acid fast stain. It is pink or red. Okay, so that is the characteristic of nocardia. So nocardia may be recovered from standard non-selected medium, but it will take them at least a week or long for you to recover them. So they're generally found worldwide in the environment and mostly affecting people who are immunocompromised, meaning people with weak immune system. So these are the significant nocardia species. So they are slow grower and may take 3 to 6 days to grow. And the common denominator of the patient's profile is that they are immunocompromised. They are rare, but they would also have a very high mortality rate. So mortality rate of about 40%. So 4 out of 10 patients will usually die from nocardia. So what are the different nocardia species? So we have nocardia, Syria, Georgica. Let me pronounce that again. So nocardia, Syria Georgica. Syria Georgica. And the other one is nocardia porcinica. Okay, so it's tongue twister, no? So they would have these two would cause confluent bronchopneumonia. So usually characterized by necrosis with abscess, and there's no sulfur granules. The other one is nocardia brasiliensis. So there would be cutaneous infection termed actinomycotic mycetoma. So actually, there are two types of mycetomas, no? So it could be bacterial or it could be fungal. If it's bacterial mycetoma, we call it actinomycotic mycetoma. So actinomycotic mycetoma is an example of bacterial mycetoma. So it is characterized by trauma to skin. There would be, so trauma to skin means that there is a traumatic implantation, and there would be major destruction of tissues and bones. So this illustration here is an example of actinomycotic mycetoma or the bacterial mycetoma. So what's the difference? For bronchopneumonia, you will not be able to see sulfur granules. But for actinomycotic mycetoma, you'll be able to see sulfur granules. So this would be the basic difference. Okay? So let's talk about the virulence factor. So another illustration here of actinomycotic mycetoma. I'm so sorry to ruin your lunch. But nonetheless, the role of factors such as toxins and extracellular proteins is unclear. So the clinical infections occur either from inhalation of organisms, that is for the bronchopneumonia, insole or dust, or associated with impaired host defense due to underlying disease or compromised immune system. So usually diabetic is at risk of getting this one, or people with weak immune systems such as patients with AIDS. Mortality rate is high because of the compromised immune system. Survivors often have a significant tissue damage. You may recover, but the damage, such as this one, is already irreversible. So, disseminated infections will involve brain abscess. So, these are usually reported. So, brain abscess. So, this is an example of a lung infection, a chronic pulmonary infection. So this is a confluent bronchopneumonia. So you can see, as you can see, no, the X-ray of the lungs is hazy. So kapag ganito ka hazy yung X-ray, meaning to say, there's so much happening in the respiratory system and the breathing of the person is actually being affected. So it's a chronic infection. Usually it lasts for months up to several years. And because of the chronicity of the infection, dissemination to other organs may occur. So, unlike the acute pulmonary infection, which could be measured in weeks, so pulmonary infection is usually seen in patients with underlying immune defects. Okay. So let's talk about the nocardiosis pulmonary infection. So at first, there would be initial lesion in the lungs. So there would be often focus of pneumonitis that advances to necrosis. So this is an example of an autopsy from the lung of the patients who have had tissue necrosis. So tissue involvement may occur, resulting in the damage. So little inflammatory response or scarring may also be possible. So the specimen of choice for us to diagnose this particular type of infection would be sputum. So we would expect the sputum to be thick and purulent. But take note that there won't be any sulfur granules upon staining. So usually kasi sulfur granules are seen in bacterial mycetoma. So masses of filamentous organisms bound by calcium phosphate is possible when observed under the microscope, such as this one. Okay, unlike nocardiosis cutaneous infections, here you'll be able to see sulfur granules. Yan, so merong active for sulfur granules. So It occurs after inoculation of the organism into skin or cutaneous tissue. So as what I've mentioned, there is what you call traumatic implantation. So there is what you call traumatic implantation. So infection begins, as a localized subcutaneous abscess, so referred to be as the actinomycotic mycetoma. So as what I told you, there are two types of mycetoma. If it's bacterial, we call it actinomycotic mycetoma. If it's fungal, we will call it eumycotic mycetoma, which you will be discussing in mycology next sem if after you have passed bacteriology. So since the infection may reach up to the subcutaneous tissue, okay, there would be destruction of tissues and even the underlying bone. Kasi nga, the destruction goes deep into the bone. Okay? So, nocardiosis cutaneous infection, as the infection progresses, burrowing sinuses will often to skin surface and drain pus. So, ibig sabihin, merong lalabas na pus dito. Ito yung mga draining pus. May mga lalabas na pus dyan. Okay? Pus may be pigmented and contain sulfur granules. So, this is how the sulfur granules would look like. Okay? So, granules often appear yellow or orange with distinct granular appearance. Okay, so another example of the sulfur granules. So, we collect sulfur granules from draining sinus tracts. So again, draining sinus tracts. So this could be the possible site of draining sinus tracts. Kawawa talaga yung mga patients na merong nocardia. So the specimen of choice, if you're suspecting pulmonary infection, syempre, the specimen of choice would be sputum. But if you're suspecting cutaneous infection, actually they aspirate from skin or abscess. So we'd expect the organisms to be gram-positive, beaded branching filaments. So they are beaded. So this is an example of beads. Beaded branching filaments. Cocobacillary bodies may also be present, and presumptive ID would be filamentous, as what shown here, branching isolate, and they are partially acid fast. So in acid fast, we are usually using acid decolorizer, and it has to be weak, containing only about 0.5 to 1% sulfuric acid. And this is how they look like, again, on wet mounts. Sorry, hindi pala to. This one is the gram stain. Okay. But there's also possible that you can do wet mounts. Tissues and pus from the draining sinuses are specimen of choice. Specimens may be seen when specimen or culture isolates are prepared properly. So ano ba yung wet mounts? Yung wet mounts, ibig sabihin, wala akong picture dito. But yung wet mount is yung when we get the specimen from the sinus tract and we add NSS and directly observe them under the microscope. For cultural characteristic, no specific growth factor is required, meaning to say, since they are fungi-like, they can grow on non-selective medium, and they can even grow in Thyer-Martin medium. Thyer-Martin is the same culture medium that we are using for the isolation of Neisseria. And they appear chalky, ayan, no? Chalky, matte, velvety, and powdery, and the color is whitish, yellowish, pinkish, orange, pink, tan, and gray. They may have dry, crumbly appearance. So breadcrumb colonies. So this is how they describe the colonies of nocardia, breadcrumb colonies. And upon using the dissecting microscope, you would actually, it may reveal the presence of aerial hyphae. So actually, yung hyphae sa fungi lang makikita yan. But since this bacteria may resemble fungi, so it's actually provide the first clue to identify the colony as nocardia. yung aerial hyphae upon using dissecting microscope. Dissecting microscope or scanning microscope. So another example of the colonies of nocardia. Okay, so nocardia is different from mycobacterium. So they are branching, fine, intertwining, delicate. They are extremely variable. So some of them are beta hemolytic, wrinkled, appears dry, chalky, whitish, crumbly, kaya nga sinawag na breadcrumb colonies. Unlike mycobacterium, which is strongly acid-fast, nocardia is slightly acid-fast. Nocardia can be positive with nitrate and urea, and they are lysozyme resistant. So that's how they are differentiated with mycobacterium. So again, mycobacterium is strong acid-fast. Nocardia is just slightly or partially acid-fast. Ito pala yung wet mount. So this is how they look like in wet mount. You'll see crushed granules. Ito yung mga crushed granules in wet mount. So under the microscopy, you'll see gram-positive branching filaments from direct smear from sputum or aspirated materials. So they may show beaded or beading appearance after 3 to 6 days on culture media. Nocardia can also be identified using the paraffin bait test because only nocardia can break down paraffin. But this is the wet mount of nocardia. So ito yung crushed granules. Another example of crushed granules for nocardia. And then this is the branching filament of nocardia. And then this is the crushed granules. Okay, so sana malipat ko sa next slide. Yan. So, nocardia can hydrolyze casein, tyrosine, xanthine, and hypoxanthine. Other substrate and that can be hydrolyzed by nocardia are aryl sulfatase, gelatin, and carbohydrates. And another way of identifying them is through antimicrobial susceptibility profile. And in high-performance liquid chromatography, they would also have a fatty acid, and Malbitov is another way of identifying them. So, treatment. Is there a treatment? Yes, there is a treatment, but unfortunately, even if you have treated nocardia, the tissue damage is already irreversible. So, you have to drain and do surgical intervention along with antimicrobials. Antimicrobials profile may vary. So sometimes they are resistant to penicillin, but they are susceptible to sulfonamides. So since these are bacteria and even if they do resemble fungi, antifungal agents have no effect. So kailangan pa rin ng antibiotics. Nocardia is present along with other organisms including fungi. So there's a possibility na merong multiple infection yung patient. So proper testing and antimicrobial treatment therefore is essential because it's very important that doctors would need to differentiate whether it's bacterial or fungal. Kasi kung bacterial, doctors will be giving antibiotics. If it's fungal, doctors would be giving antifungal. Other actinomycetes would include Actinomadura, Streptomyces, Gordonia, Chocamorella, Rhodococcus, and Tropherima, Whipple, or the causative agents of Whipple's disease. Okay, so I hope that you'll be studying this table. And we will now be proceeding to the last topic for this morning. And this is the spore-forming, non-branching, catalase-positive bacilli. So obviously, you'll be able to see the spores here. Okay, so first in our list would be the Bacillus species. So they are gram stain positive, some are gram variable, so they are aerobic, and some are facultative anaerobic. So they have endospores which will make them highly resistant to heat, drying, and chemicals, and they are widely distributed in soil and in the environment. So Bacillus species are metabolically diverse, and they can grow well in chocolate agar plate, but not on chocolate agar. They can grow well in sheep's blood agar, but not on chocolate agar. Most species are non-hemolytic and catalase positive, and many members of the species are regarded as laboratory contaminants. And perhaps the most common laboratory contaminants is shown here. This is an example of Bacillus subtilis, the most common laboratory contaminants. So many laboratories do not identify Bacillus up to the species level, but there are two medically important species. Later on, we will be discussing them. One of the medically important species is Bacillus anthracis. So the common name of Bacillus anthracis is the anthrax bacillus. It is encapsulated. In fact, they have D-glutamic acid capsule, which can prevent phagocytosis. And anthracotoxins have three proteins, namely the edema factor, the protective antigen, and then the lethal factor. Vaccination is available, particularly those people who are at high risk of exposure. So people who are high risk of exposure are those who are working in the farm, particularly with sheep or with cattle. Um, Bacillus anthracis epidemiology. So this is common where livestock is present. And eventually, they are also common when vaccine is not being used. So human infection is actually accidental when you ingest the spores or occupational exposure. And recently, we also have the so-called bioterrorism. So after 9/11 attacks, many bioterrorists would be placing the spores of anthrax in the mail. So when you open the mail and powder will puff out from the mail, it could contain the spores of anthrax. So there are three major forms, but now they have identified the fourth type. So cutaneous. So this is the cutaneous anthracis, and then this is the intestinal anthracis because this is the villi of the intestine, and then the pulmonary anthracis. Anthrax, the pulmonary anthrax. So they are using anthrax as the agent of bioterrorism. So the most notable was during 2001 after the 9/11 attack on the World Trade Center. So many senators in the US, in New York, or even in the White House have received mails. So pag open nila ng mail, it contains a message such as, "You cannot stop us. We have this anthrax. You die now. Are you afraid? Death to America, death to Israel. Allah is great." So we have nothing, of course, against Muslim because only few extremist Muslim groups are the ones doing it, but generally Muslims are good people. Okay, so when they open the mail, um, powder powder goes out. So can you just imagine? No, you're reading it, but it's too late already because you have already inhaled the powder that contains the spores. Okay. Bacillus anthracis clinical infection. So I told you a while ago that there are three types, no? So the skin, we call it cutaneous, and then the pulmonary is through inhalation. Then intestinal anthrax is when you ingest something, when you ingest the spores accidentally through unprocessed milk or contaminated meat products. But there's a fourth way of having an anthrax. It's injectional anthrax. The fourth form of infection is now recognized because sometimes when you inject something in our skin, spores will also be injected. Usually, for those who are doing heroin abuse, so they're injecting heroin into the vein. At first, they do not observe hygienic or aseptic technique in doing such a thing. So spores will be injected as well into the skin. So that particular thing would result into injectional anthrax. So Bacillus anthracis will cause cutaneous anthrax. So it occurs when wound become contaminated with anthrax, and it will be acquired through skin cuts, abrasions, and insect bites. So it constitutes about 99% of anthrax cases, and most of these happened in the farm. Okay, so there would be a small pimple or papule that appears at the same site of inoculation 2 to 3 days after the exposure. So there would be a blackening, a black ulcer. So we call it the scar formation. So the scar formation is actually found in anthrax. Okay, so the cutaneous anthrax should evolve into lesion in a ring vesicle, erythematous ring, and small dark area in the center. So there is a black necrotic center. So this black necrotic center is called a scar formation. So a scar begins to heal 1 to 2 weeks, and lesion will dry and eventually falls off, leaving a scar. Usually it is a localized infection, but it can spread. But there's no systemic symptoms present. So fortunately, there's no systemic symptoms present. Okay, so this is the first type of anthrax, cutaneous anthrax. So the second type is called pulmonary anthrax. So sometimes it is known as the wool sorter's disease. So those who are working with sheep, sometimes they harvest the wool from the sheep kasi this can be made into clothes, di ba, wool. So people who sort the wool, we call them wool sorter. And as they sort the wool, they can also inhale the spores, causing them to have pulmonary anthrax. So it's acquired through inhalation of the spores. So there would be mild fever, fatigue, and malaise for about 2 to 5 days after exposure. So people would be experiencing symptoms similar to flu or common colds that would last for about 2 to 3 days. So there would be sudden respiratory distress, disorientation due to very low oxygen level, coma, and death. And it may last in less than 24 hours from the onset. Then people will die afterwards. So the third type of anthrax is the intestinal anthrax, the gastrointestinal anthrax. So we usually get it after we ingest spores via contaminated raw meat. So it inoculates into a lesion on the intestinal mucosa. As you can see, the intestine here would have several lesions. So it is characterized by abdominal pain, nausea, anorexia, vomiting, and sometimes bloody diarrhea, more likely to be fatal but less likely to occur than cutaneous form. So it's difficult to diagnose and it's very rare, and it only accounts for less than 1% of the infections. And then the fourth type is the injectional anthrax. So mostly affecting the soft tissue with skin popping or other form of injection drugs use. Because when we inject drugs, sometimes we also inadvertently inject the spores. So unlike the cutaneous anthrax, it doesn't form the black scar, but it can cause necrotizing fasciitis, organ failure, shock, coma, and meningitis, and it has a very high mortality rate. So do not be a drug addict. So is there a vaccine available? Yes, there is a vaccine. So about 5% of patients with anthrax develop meningitis. So more commonly seen in the inhalation and injectional form. So meningitis is not common in intestinal and cutaneous. It rapidly develops, so patient will be unconscious and death will follow afterward. 1 to 6 days after the initial exposure. So napakabilis, no? So effective vaccine is available. So it is made up of cell-free culture filtrate of bacterial proteins absorbed into aluminum salt, and aluminum salt serves as the adjuvant, which can enhance the immune response. So there is an anthrax vaccine absorbed available. So commercially, it is known as the Biotrax. That's the brand name. So this is how Bacillus anthracis look like under the microscope. So under the microscope, they are characterized by large square-ended gram-positive or gram-variable rods. that sometimes they are single or appears in chain that looks like bamboo. So spore stain on direct samples would yield a positive result. For the colonies, you would notice that they are non-hemolytic on 5% sheep's blood. So this is how they look like. Grayish-white irregular edges. And when looked closely under the scanning microscope, it appears like medusa head or beaten egg whites colonies. Spore stain is negative from the culture. So this is an example of spore stain. So usually we are using malachite green when staining the spores of Bacillus. So this is how the malachite, ayan, makita nyo yung green. So that's positive for spore stain. Okay, so whenever you are working with Bacillus anthrax, so you do not want to inhale the spores. It's very dangerous. So you work under biosafety hood when Bacillus anthrax is positive or suspected. So the organism is catalase positive. They can grow aerobically and anaerobically. They can ferment glucose and does not ferment other sugars. Okay, and they can produce the cutaneous. So another sensitive way of identifying Bacillus anthracis is using the direct fluorescent antibody test because it confirms the presence of antigen. Ano ba yung antigens na to? Cell wall polysaccharide and capsule. So the organisms, since they do not have flagella, so we expect them to be non-motile. They can grow in high salt concentration and in low pH. So aside from Bacillus anthracis, there's another one that is medically important, and that is the Bacillus cereus. So Bacillus cereus can be involved in food poisoning. Okay? So to differentiate them, Bacillus cereus can cause hemolysis and blood agar plate. Um, they can be positive uh, in motility, but they are resistant to penicillin, unlike Bacillus anthracis. Bacillus cereus can produce the citines, and they may ferment salicine, and then it can even grow in agar that contains penicillin. They can hydrolyze gelatin, and they can be grown in phenylethyl alcohol agar or the PEA medium. So the medium that we are using for Bacillus anthracis, sometimes timba, and for Bacillus cereus, we are using the flat medium. Okay, so before we go further, kasi later on, after we discuss Bacillus cereus, after we discuss Bacillus anthracis, we will be discussing Bacillus cereus. But let's discuss first the treatment for anthrax. So resistance can occur due to beta-lactamase production. So penicillin should not be used alone. So usually we can use other antibiotics such as tetracycline, fluoroquinolones, and chloramphenicol. And CDC recommends using ciprofloxacin and doxycycline intravenously. So let's talk about Bacillus cereus. Bacillus cereus um, can cause food poisoning, and it is actually due to the distinct enterotoxins. So the food poisoning can be emetic form, and the other one is diarrheal form. So yung diarrheal form is associated with meat, poultry, and vegetable pastas. Incubation period is 8 to 16 days, and abdominal characterized also by abdominal pain and diarrhea. So the fever is uncommon, and it resolves within 24 hours. So indistinguishable from Clostridium perfringens. Kasi nga, this one is diarrheal form. So the other one is the emetic form. So if this is the diarrheal form, and we have the other one, which is emetic form. So emetic form means that there's no diarrhea. only nausea and vomiting hours after ingestion of contaminated food. So diarrhea is present in 1/3 of those affected, but it's not really common. Unlike in diarrheal form, there would always be diarrhea. So it is associated with fried rice. So there would be abdominal cramps and vomiting. The incubation period would last for about 1 to 5 hours and resolves in 9 hours. So the diarrheal form is caused by heat labile toxins, enterotoxins. So ibig sabihin ng heat labile, if you will be heating the food, the toxin will be destroyed. But there are some foods that are not really being heated, such as yung mga salad or yung mga cold cuts, di ba? Hindi naman sila hiniheat. So the chances of us getting diarrheal form in this food would be very high. That is due to heat stable toxin. Meaning to say, even if you heat the food, the toxin remains viable. Kaya nga, kahit na yung fried rice, di ba, hinit natin yung rice. But still, the toxin remains viable. So there are two distinct enterotoxins. Yung sinabi ko, yung heat labile and heat stable toxins. So again, to differentiate the type of enterotoxin, for the diarrheal form, the incubation period is 8 to 16 hours. Emetic form is 1 to 5 hours. Diarrhea is very common in diarrheal form, but it is not common in emetic. Vomiting is occasional in diarrheal form, but very common in emetic. The duration of illness would be longer for diarrheal form. Ito nga yung makikita natin, no? Meat products, soup, vegetable puddings, and soy sauce, sauces, sorry. And then dito man sa emetic form, fried or boiled rice. So the enterotoxin here has higher molecular weight higher in in diarrheal form, and it is heat labile. Ito naman, it is heat stable. Okay. And aside from food poisoning, Bacillus cereus can also cause infection. This is the most common non-gastrointestinal infection. So this infection is called endophthalmitis, panophthalmitis, and keratitis. So usually there would be abscess. So there would be poor visual outcome when infection is in penetrating the ocular, particularly in trauma cases. So natusok yung mata, literally. So rare infections in immunosuppressed and post-surgical host. So pwede sila magkaroon ng meningitis, septicemia, endocarditis, and osteomyelitis. So how do we identify Bacillus cereus? In, in blood agar plate, they are beta hemolytic and they would have the frosted glass-appearing colony, katulad nito. On gram stain, they are spore-forming gram-positive bacilli. They are motile and able to ferment salicine, and they are lecithinase positive. So another example of Bacillus cereus on blood agar plate. And treatment, they are resistant to penicillin. They are resistant to other beta-lactam antibiotics except for carbapenems. And therefore, we have to treat patients with vancomycin and clindamycin with or without aminoglycoside. And other species of Bacillus include Bacillus subtilis, which is the most common laboratory contaminants like Cuniformis, Circulans, Cumulus, Syphyricus, and baka nalimutan nyo na yung Bacillus thuringiensis. Ito, ginagamit to sa mga genetically modified organisms. And then, baka malimutan niyo yung Bacillus thermophilus, yung ginagamit natin na indicator for autoclave. And that ends our aerobic gram-positive bacilli. So thank you very much for listening with me.